Okay. Just a sec. Okay. I think mix it. Okay. All right. Thank you. Welcome for joining for the problem solving session 8 of Animal Physiology, which is a course, as we know, offered by NPTEL. And we will be continuing our discussion on a topic today, which was done in the very first few classes for this course. And then we will switch our gears and we'll do completely different set of systems, which is basically smells and sound. So auditory system or hearing system, and olfaction system, which is basically the taste or sorry, the smell, right? So we will be focusing on this, and like always, the session will begin with a recap for what was done in this week and the topics that were covered, just to give you a brush up if you can remember the notes. And then I have a lot of questions for you so that you get well versed with the topics, different kind of questions, in the sense different usage of words, so that whenever you are revising for your exam or whenever you are revising the whole course, my videos serve as a revision track for you. You can come back to these videos and do these same questions so that you get a better idea of this topic. And these are a lot of questions. Hopefully we'll cover all of these today. But like from the past few sessions, I am giving all the questions out so that even if we haven't discussed in the session, it's there in the uploaded video. You can always use it for your reference, right? Okay, so let me begin with this session today and try to show you that what all did we do in this week 8. So week 8 started with stretch reflex circuit. We already know about stretch reflex arc from the very first few classes. We know what is stretch reflex. In very simple terms, is the knee-jerk reaction. We know already about that. But what we are more interested is in the connection between your stretch reflex and your so the nitty-gritties of how does it really translate from detection of the signal from the muscle to the action of the muscle. How does that entire electrical and the muscular system work, right? If you remember it, this was an add-on to the previous things that we had discussed. And in reflection or in relation to these, we had the neuromuscular junction, where we actually saw how does this information get converted into the neurotransmitter, and from there the action potential in these skeletal muscles. And with that, we ended this topic. Yes, you can please post your doubt in the chat box. I will come to the chat box and I will read and let you know, if I can help you with this. So please put it in the chat box, your doubt, so that in the meanwhile, I'm just reading the screen slide down. Okay, and the other two systems that we discussed in week 8 was hearing system and olfaction system. So you have missed the submission of the assignment. Okay. Well, that's a hard, then actually you need to submit it on time because you're graded on them. Yeah, so you need to submit a certain proportion of assignment. So make sure that you submit all the other. This assignment is gone. You're not graded on this, but I'll just remember that yeah. So just make sure that you don't miss out on any other assignments, because the assignment marks are also taken up for your eligibility in the final exams. So don't miss out, my suggestion to you. And yeah, just remember that all the other assignment, barring the one that you missed, you please remember to fill it on time. Yeah, and just be punctual. That's it. Don't stress about what's gone already. Okay. So let me give you a brief revision of what you did about stretch reflex arc. In very simple terms, I'm pretty sure, considering the amount of time we have dedicated to discuss about stretch reflex arc, we know about it probably way too much. But just for your revision, what does stretch reflex arc means? It's basically the entire process of your muscle sensing some change in your regular muscle posture and then bringing it back to its original structure or posture. Now, when I use these type of words, I hope you don't get confused, because as I have been mentioning in the previous class also, terminologies matter in this course. Even though you don't have a really subjective topic, but do remember when you give your exams or when you are using this subject for any other exams, the technicalities matter. So words like contraction of muscle, words like relaxation of muscle, stretching of muscle, these mean something and you need to remember it that way. So hopefully I will use those words today in the lecture, and that should give you a cue if you haven't yet thought about it in that way. Okay, so let me reiterate. So stretch reflex arc is basically a simple and rapid reflex. Reflex is anything. So reflex is a common term that we use in our day-to-day activities. This is very reflex of you, reflexive action. We feel something. For example, in very simple term, you have, so there are two reflexes which I am sure pretty much everyone knows. One is touching a hot surface and then removing your hands, and second is someone hitting on your like below your knee, and you are jerking your knee. Your knee automatically kicks forward, right? So those are the two kind of reflex action that everyone knows about. But there is how do I say, there is a difference in the mechanism of these two reflexes. Okay, and we are going to particularly tackle about the patellar ones, which is basically the one which is involves a knee-jerk reaction. I'll tell you what I mean by that. But basically, this is a reflex arc, which basically involves your the ultimate goal of a stretch reflex arc is to maintain your muscle length and posture. So any change in your muscle length or muscle posture uh, initiates the stretch reflex arc. Okay, now what all do you involve in stretch reflex arc? Sorry about that. You have first the receiver. So it starts from here. You have the receiver, which is generally a muscle spindle. Your muscle is the one which receives the information. What information? It uses the sensory receptors which are located within the muscles, and then you detect the muscle stress. So basically any length changes. Here, for example, it is basically like let's say you are touching a hot object or your knee is being hit by some hammer. So all these different kind of information, it the muscle stretches in response, and this muscle stretch, which is a change in your muscle posture, is sensed by, if you remember, intrafusal muscle fibers. Remember the term intrafusal. We had discussed this in the way where I told you about intra and extrafusal. Extra, I told you to remember as an outside perspective. We talked about efferent and efferent. Efferent is coming inside, efferent is going outside. Anything that goes outside is extra, so you can remember it in that way. So you can make your own cues and shortcuts to remember stuff, but just remember that you need to relate the correct terminology with the respective processes, right? So the one who senses, the muscles which sense are the intrafusal muscle fibers. They have something called type 1A neurons and type 1A afferent fibers. So they will detect, and then starts the afferent pathway. Afferent is basically something that takes it forward. So the muscle spindle now contacts the spinal cord. So this is your afferent network going all the way to spinal cord. Now, spinal cord is here called the integration center, which is basically processing all these different information that it is getting. And from there, whatever decision it takes, decision like the forward kick of the knee or the removal of your hand from the hot object, that decision is then passed on to the efferent neuron. Efferent is something that is coming out. Efferent is basically something that emerges out. So efferent neuron, which comes out of spinal cord and which takes it to the, so this efferent neuron is basically the motor neuron. Motor because it is going to do the activity, the movement activity. So it's the motor neuron, and it again takes it to the muscles because it is a muscle that has to move. But this time the muscle is called effector muscle, which is generally the fibers are the extrafusal fibers. So all the terminologies taken together cumulately, if you just once remember the terms and connect them, then it should be easy for you to remember the next term. So all the words that I just told you from the left figure, which was motor, muscle spindle, and 1 alpha, and all of these are, if you remember one pathway, it should be becoming easy for you. Now, I told you two pathways, which is, or two reflex action. One reflex action, I have submitted assignment at once time, so it can be considered for final exam or credit. It will be considered. All your submitted assignments or all your assignments are considered for your exam. Okay. So if you have submitted in the portal, or if you have uploaded it, and basically what I mean is, if you have from your side responded to the assignments, you have uploaded and answered it, that will be considered. At one time, if let's say you have given two, it's what I'm expecting that you have done two at once. So all the assignments that have gone together or from your account will be considered for the final exam. Okay. Yes. So coming back to this, yeah. So I told you about two reflex actions, right? One reflex action is me, welcome, no worries. So one reflex action is me touching a hot surface, removing my hand. Second, someone hitting with a hammer just below my knee and my knee moving forward. These are reflex actions. All these pathways that I've just told you about, the intrafusal, extrafusal works. But there is a basic difference between these two reflex actions, and that is what you need to remember. The difference is in the way the synapses function in these. Okay, you have a monosynaptic reflex and you then have a polysynaptic reflex. Now, what is a monosynaptic reflex? As the name suggests, monosynapse, one synapse only. Now, what is a one synapse? What are you discussing about this here? So mono means one, and that basically means there is a direct communication between sensory and motor neurons. So your sensory neuron takes it all the way to from spinal cord, and from there it passes directly to the motor neuron. So in this whole reflex action, you only have the communication of sensory and motor neurons. Okay, this is what is stretch reflex. The other reflex that I told you is not a stretch reflex, that is a withdrawal reflex. That is basically touching of a hot surface and you are removing your hand. You are removing your hand. That's a withdrawal reflex, right? So what happens there is, why it is called polysynaptic reflex. What is polysynaptic? Multiple synapses. Where does the multiple synapse comes? It comes when your sensory neurons or sensory, between the sensory and the motor neurons, you have the interneuron, as you can see here. So this is your sensory neuron. This passes the information to interneuron. Interneuron takes the information to motor neurons, right? So poly means multiple neurons, and mono means just two neurons, just one synapse in between them. Now, what is the inherent difference between these? Why do you need these two type of difference? Because remember, we keep telling about we need more synapse so that information is there and this and that. Then why do we have these two differences? Stretch reflex has only monosynaptic and withdrawal reflex has polysynaptic. That is basically dependent on the speed of the conduction. So in case of your monosynaptic, where you have only one synapse to cover, your activity or your final outcome, which is basically stretching your muscle. So the knee jerk reaction is an example of stretch reflex arc. So there you need to respond very quickly, right? And that action takes place very fast, and it's a very simple system. Like there's no complexity here because there is one neuron to the other neuron, right? But what happens in poly, because there is multiple junction to cross, it's a slower process. So your removal of your hand, we might not realize it, but if you think about it, the knee jerk reaction and your removal of your hand from your hot surface, they lag in time. Your removal from your surface of hot surface basically slows down, and that's because you have so many information all need to cross the synapses of interneurons and then reach to the motor neurons, right? So that is a slower process. What does it do in hot or cold surface? You have multiple information gathering, so your information gathering is more, so the process is slowed. While in the case of stretch reflex, where you just have to, someone is basically, so what happens in your knee jerk reaction is, let's say this is, for example, just imagine this to be your knee and someone is hitting with your hammer here. This is what is happening, right? So here you have the quadriceps. These quadriceps start basically stretching, right? So they are responding to the hitting that they're getting. This needs to go very fast into normal posture, right? Because it is starting to stretch. So you just have these one neurons pathway, which basically lets the knee muscle or the muscles here to contract. As soon as the muscle contracts, this moves ahead. This is what you will see in the knee jerk reaction. And that's a very quick reaction. That's why whenever in normal terms, whenever you say, oh, you reacted very quickly, you say it was a knee jerk reaction. It was very impulsive. So knee jerk is very fast. While let's just imagine you are touching a surface which is very hot. You touch, you instantly remove. That's also in millisecond, but that is still slowed process as compared to the knee jerk reaction. If you observe it, if I mean, henceforth, if you see this kind of reaction, you will realize that the timing between these two is different, and that's due to the polysynaptic and monosynaptic nature of these two processes, right? So we also did this in the week 8, and then this is something that we had already done in the first few weeks. The types of muscles that are involved in your skeletal stretch reflex arc and the type of neurons that are involved in a stretch reflex arc. We already know about extrafusal fibers. We already know about intrafusal muscle fibers. Extrafusal muscle fibers are outside, so they are contractile because they will contract. They will be the one who will bring the skeletal muscle back to the contracted position. They will generate force and movement. Right. So what are the key terms that you need to remember? In extrafusal muscle fibers, there will be force generation and movement. And which are the associated neurons? Always remember, how do you remember? You can just connect these two topics and remember. The muscles and the neurons go hand in hand. Which muscles are these and which neurons are there? That's how you will. Oh, sorry. I had a question here. Ma'am, in the stretch reflex, how does it decide to go back to? There is no interneuron in stretch reflex arc. There is no interneuron. That's why it's monosynaptic. Interneurons are in withdrawal system, right? And how does it reflect to? Because it's so, just imagine this is muscle, it is stretching. The whole idea is to remove the, if it is stretching, the whole idea is to bring it to the original position. So if it is stretching, it comes back to the original position by contracting. It is stretching, it is increasing in size, it is contracting, and it's coming back to the original size. That's the whole agenda, and that's the whole purpose of stretch reflex arc. Interneurons are not part of stretch reflex. That's why it is monosynaptic. The interneurons are there in the withdrawal, which makes it a longer route to travel information for. So that's the difference here. There is no interneuron first. Second, how does it decide to go to original position? Just reversing the action that first gave the distortion in the muscle posture. If it is stretching, it is contracting. That's what it's that. That is why the name stretch reflex arc. Something is stretching, and there is a reflex to this entire stretching of muscle to bring it back to original position. The name of the entire process describes what is happening. So it is stretch reflex arc. That's how you can remember what's happening. That way you can never forget. This is a stretch reflex arc. So your muscles are stretching, and it is reflection of, or it is reflexive of this stretch in the muscle. That's acute for you to remember. Yeah. And about these topics, you need to remember your extrafusal muscle fibers, intrafusal muscle fibers, and you need to remember which neurons are associated with them. Because the extrafusal fibers are the extrafusal muscle fibers are the ones that do the generation of force and movement. They are associated with the motor neurons. Okay, so and the and the intrafusal muscle fibers, these are associated with the motor neurons. Which one? Gamma motor neurons. And we'll see why. Just give me a second. When I move to the next slide, I will show you why a motor neuron here as well. And the sensory neurons are also part of intrafusal muscle fiber because they are the one who sense it, right? Okay, now let me bring you to this part here. Ma'am, which book get this content? Okay. So these are very well-researched topics. I mean, you can simply search it up. You will have a lot of information on stretch reflexer. This is a very easy topic. But if you still want about physiology in general, there's a book called Guyton, which will help you with the these physiology. But that's medical physiology mostly, but it will help you get the idea of all these topics that you are studying. So it's called Guyton Medical Physiology or something like that. But the book's author, I will type it on the chat box just if someone is interested. Yeah, so you can use this book for your reference. But do remember they're way too much into detail. So you might just want to read the gist of it or overall theme of it. Okay. Yes. So like I was saying, you have alpha motor neurons, you have gamma motor neurons and sensory motor neurons. Now you can already see where are the sensory motor neurons. They are with the intrafusal muscle fibers. Makes sense. Intrafusal muscle fibers are supposed to sense and the sensory neurons are also there. Now, you have two type of motor neurons, alpha motor neurons and gamma motor neurons. You will see one of the motor neurons is with extrafusal muscle fibers, which is very easy to understand, of course, because they are the ones who are generating movement. So the motor neurons are with them. So they're the alpha. You can remember it's the primary role of extrafusal muscle fibers and alpha is the one which is associated. Now, what is this extra gamma motor neuron doing? They are in the intrafusal muscle fibers. So what are they doing? Their primary role is to adjust the sensitivity of the muscle spindle during contraction. So when your muscle is going back to its original position, which is contraction, at that time it is in role when it is trying to adjust the sensitivity. So it is basically helping in the regulation of the muscle spindle, right? So it has a role in the muscle spindle. uh, After finishing lecture, yes. I hope I so all my lectures are available. In fact, all the lectures which are taken as TS session for NPTEL courses, you have the YouTube videos, the sessions YouTube videos, and the PPTs that are there, which will be uploaded on the link provided to you. So you will find both the PPTs that I'm using for class as well as the recorded lecture for the class in the site, in the link that is provided to you, which is also there in uh, the links that will take you to the master folder, which has all the previous PPTs as well. Right. Yeah, so that is what is alpha, that is what is gamma. So gamma is when the muscle is in action. That's why it's in intra, because intrafusal is the one for sensitivity, and that's what needs to be maintained for the next round of cycle. And sensory goes with the sensory neurons. Okay. Now, somebody asked me about interneurons. So you will not find interneurons in monosynaptic stretch reflexes. Okay. Because I told you it's monosynaptic. When it is polysynaptic, then you have interneurons, and the function of interneuron, as the name suggests, it is between the sensory and the motor neuron. It is for the relegating of information in the whole pathway. Okay. Okay. Now, fantastic thing that we have understood. Oh, there is a stretching, or there is some muscle distortion. That muscle has taken it into neurons, so it has become now a neural problem. When it comes back to your muscles with the neuron, how does it go? So how does the neuron give an information for the muscle to contract? I hope when you ask me about the decision to go back to the original position, it's basically the reversal of stretching. But the mechanism of reversal, or basically what I mean is, how does the muscle contract under the information received by neurons, is because of the neuromuscular junction, because we have already studied about this, right? That there is a junction. So this is your muscle, okay? And there is a nerve ending. So these nerves, sorry. So this is the nerves, right? Let's bring alpha and motor neurons. So this neuron is coming here. This is a muscle. How does this neuron tell something to the muscle? How does the neuron say, hey, you have to contract because you are not in the correct position? So there is an entire mechanism behind this, and this is why neuromuscular junction become very important. Okay. So neuromuscular junction are basically the ends of your alpha motor neuron. Makes sense because they have to give the information to the muscle fibers. So these are at the end of your alpha motor neurons, and you have the action potential. We know in neurons you have action potential which is moving towards the end, and we have also discussed that when you are looking at the neuromuscular junction, you have the acetylcholine transmission, right? So your neurons have this action potential which comes here. The vesicles which contain your calcium, they break open. So calcium mix, then your vesicles also, because of the calcium, excessive calcium, the vesicles which consist of your neurotransmitter fuse with the cell membrane and they break off and come into this cleft. This is called synaptic cleft. Now, in your synaptic cleft, what is released? Acetylcholine. Now, what does acetylcholine do? Your acetylcholine will bind to its receptor here, okay, on the muscle fibers. Now, as soon as your acetylcholine binds to nicotinic acetylcholine receptor on the muscle fibers, what happens is that your because there is a binding there, the sodium channels open up, and the sodium channel open up leads to something called depolarization, if you know, because as soon as the sodium channel opens up, we know the entire process of action potential generation, which begins because of movement of sodium. We have dedicated an entire probably 2-3 classes to just understand how does this entire arc take place, right? How does this movement from resting membrane potential go to depolarization? Here also in the muscle, as soon as your acetylcholine comes in, on its receptor, immediately sodium is released. Sodium movement takes place from the membrane. The same thing happens, your action potential is generated now in the muscle. Now, how does this muscle or how does this excitation because of action potential relates to contraction? Beautiful process and very simple to understand. It should be very easy once you just visualize it. Okay, so this is your neurons, synaptic terminal. Your acetylcholine has come here. Your acetylcholine binds to its receptor. Now what happens? Now, as soon as your acetylcholine is binding to the receptor here, you have sodium ions which are, or sodium gate ion chairs which open up, and you have action potential. Where does the action potential here goes? The action potential goes to some like via sarcolemma to something called T tubules. Okay. Now, what do these T tubules do? These T tubules have something called dihydropyridine receptor. Just bear with me. First, let me just tell you once, and then I will come back and explain to you once again. Okay, so you have your sodium, you have your action potential, which goes to the first to the dihydropyridine receptor. And these receptor get activated because for them, as soon as your voltage of Na plus changes, they get activated. They pass on the information to ryanodine receptor. Now, what do ryanodine receptor do? As soon as your ryanodine receptor is activated, these receptors are present on, where are they present? They are present on sarcoplasmic reticulum. Now, what does sarcoplasmic reticulum have? As soon as your RYR and DHIR, basically these two receptor interaction takes place, your sarcoplasmic reticulum opens up and your calcium ions are released. Okay. And now, if you can just, yes. So now if you can, I'll come back to your question just a second. Okay. So now if you, how beautifully you can connect this is, where did I stop? I stopped that you have these two receptor, and this entire receptor interaction leads to the release of calcium. And calcium takes you to actin myosin. If you remember from the entire sliding filament theory, we had calcium, we had actin heads, we had myosin, we had troponin, the entire cycle, if you remember. So let me just tell you, if you can't remember, you have increased cytoplasmic calcium. When there is increased calcium, it binds to troponin C, which is on the actin filament. As soon as your troponin C is bound, your troponin C is now binding to calcium, so tropomyosin will move. As soon as your tropomyosin moves from actin filaments, myosin comes and binds to actin and pulls it towards itself. What does myosin binding to actin do? Contracts the muscle. Remember that removal of the Z line and A line and I line, A band, all that topic that we had done, that connects here. Now, what started all of this was a acetylcholine transmission at the receptor level. Sodium gated channels open, sodium moved in. We have entirely understood how sodium movement gives you the action potential, and this action potential ultimately leads to sarcoplasmic reticulum and calcium 2+, and calcium 2+ connects the sliding filament theory with the neuromuscular junction, and that's it. That's covers the entire topic of neurons and muscles. So this is how you connect it. I hope you can visualize. If you visualize most of these processes, it becomes very easy for you to understand. So I hope you can visualize what's exactly happening. And this entire process is called excitation and contraction coupling. Excitation because your muscle is getting excited because of the action potential generation, and contraction is the ultimate effect of this entire wrist activity. So you need to remember three receptors in their order: acetylcholine receptor, dihydropyridine receptor, and ryanodine receptor. Okay. All right. Now, like I said, I will not talk about it deeply because I have always questions that will help you to understand. But let me just remind you briefly what all did you do in hearing system. This is basically about ears. But before that, somebody asked me if I am a PMRF scholar. Yes, I am a PMRF scholar, and uh, that's how I landed up as a TA for this course of NPTEL. Okay, so for your hearing system, I think everyone, like even a kid, knows that uh, the organ which is used, or the organ which is responsible, is ear. Now we also know about the general anatomy of ear. We know the outer ear, we know the middle ear, we know the inner ear. What is the purpose, or what is the goal of this subject, is to make you understand these functions of different parts. So your outer ear is basically which collects your sound. The middle ear, which we know consists of the three bones, which is responsible for conductive mechanism, which conducts the entire sound and gives it to the inner ear. And the inner ear converts these into nerve impulses, which is basically what allows us to respond to that particular sound. Okay. Now, I will not talk about the other, like these structures here, which you already know, but three things that I will remind you, because there are questions, of course, you can always revise from that. If you remember, let me just try to shed light on hair cells, stereocilia, and kinocilia, if you remember them. Hair cells are basically the sensory receptors, and these are present in cochlea. Okay. And there on the hair cells, you have the stereocilia and the kinocilia. And these both are microvilli. Microvilli are the small projections, like hair-like projections, and they are basically what bends when your membrane, which receives your sound frequency, moves. So when your membrane moves, these hair moves. Okay, so remember these. Now let me tell you briefly, if you just for your recollection, what are the events in your hearing. Now let you just, if you can just see this entire structure here. So the first thing is, from the outer ear, your sound waves arrive at the tympanic membrane. This is a tympanic membrane here. The first thing is it reaches tympanic membrane, right? Okay. Next, now as the sound hits the tympanic membrane, there is movement of the tympanic membrane. And because of that movement of the membrane, there is displacement of the auditory ossicles. So these ossicles, these three bones in the inner middle ear, they move. Now, when you have basically a movement of these ossicles, the next thing is, so if you remember malleus incus stapes, right? That's the order of their arrangement. So as soon as the last one moves, so movement of the stapes at the oval window, which is here, that movement establishes waves. This is not difficult to imagine because there is something movement, there is a wave which is generating. In cochlea, you have perilymph, right? So that liquid, that fluid which is there, it starts generating waves, which is something, again, very easy to understand. In a pool of water, if you throw a stone, that will generate ripples, that will generate waves. That's exactly what is happening here. You have fluid, and suddenly you have these stapes, this ossicle coming and moving. So you have this rippling effect, this waves formation. Okay. And this is what is seen here. These are the waves that are forming. Okay. So as soon as this happens, you have this pressure waves in the perilymph of the cochlea or the vestibular duct. Now these waves distort the basilar membrane. Now, because these are waves which are moving, the membrane which is just located adjacent, they start to distort. Right? Okay, and where are they leading to? These membranes are on their way to the round window of the tympanic duct. So this is the duct, and they are on this lining here. Now, when these membranes are moving, so vibration of the basilar membrane causes vibration of hair cells against the tectorial membrane. So now on this membrane, you have these small hair-like projection. because of the basement membrane movement, these cells start bending. Okay. Now, these bending, this information about the region and the intensity of stimulation is related to CNS over the cochlear branch of different nerves. Let's not get into this. So basically, does the MIS won't vibrate? Basically, that's what I'm telling, right? So this, let me just show you once again. So the displacement of auditory ossicles is basically your bones are basically moving in response to the frequency that they are receiving from the outer ear. Right? I'm expecting those are malleus incussus tapes. So yes, the whole movement thing here is basically about their displacement from their original position. Okay. Yeah. And lastly, so the ultimate goal is all this movement will basically lead your hair cells to bend, and that bends gives it a mechanical pressure, and that leads to this relaying of information to CNS, basically central nervous system, and which is processed by the which is basically processed by the auditory cortex, and then based on the frequency of the sound, the amplitude of the sound, how quickly it is coming, how loudly it is coming, and from where it is coming. Let's say that's how we respond, right? If a sound is coming from our right, we will move towards right unless we don't want to move towards right, then we move towards the left. But that decision is based on the perception of the information by the ear. So what are the information that are decoded? Frequency, what in a particular given time, how quickly or how many times are you listening to that sound? So frequency, amplitude, the pitch of the sound, how loud and how quick. So the louder the voice, the nearer the source is to you. So that thing. And localization, which is it coming from this side more or this side more, or is it coming from the center? That localization. And then based on that, you are doing place coding, this coding, that coding, and then ultimately you have the response to it. And the response generally in our cases, let's say for example, I'm talking right now, and I am talking from a central perspective. So your ears are receiving it from both the ends, taking into CNS, and you're processing the words and the sounds that I'm talking about. If I suddenly go slow or low, then you will not listen, and you will have problem in interpreting the words. So you will try to focus more to listen to us. That's what we do, right? When we hear more, we try to focus and listen. So those are the responses, which is basically the coding, what the coding does, right? And then, of course, your memory comes into play. We have discussed about memory, taking these words, putting it into your semantics and storing it in your brain. All those will come into action. So your CNS, central nervous system, takes all of this information and process them, right? Okay. And the other system that you had looked into in week 8 was olfaction, which is basically smell. And basically, if you look into the olfaction system, we only know about nose. When you dig deeper, you will see that you have so many different structure. You have olfactory epithelium, which is here in the upper part of your nasal cavity. Epithelium is basically anything which is the covering of the structure. So here you have the olfactory sensory neurons, right? And then you have the olfactory receptor, because if you have sensory, you also need receptor neurons. So they will be the one who are having the odorant. So the receptors here basically are responsible for detection of molecule. I hope you realize that the smells that we are listening, the different kind of smell, for example, perfume or garbage or mild or strong, these are all dependent on the receptors. So you have molecules in your deodorant, right? If you look in your perfume or your deodorant, you will see extracts. Those are basically molecules, and those molecules have receptor in our nose or nasal cavity. Those receptors get activated because the molecules goes and binds to them, and you interpret that smell. Now let's say there is one deo that I like but you don't like. That's because of your perception. The same molecule binds to my receptor. I like the smell of it. You don't like the smell because when it's perceived by your brain, you kind of dislike it. So repellent and attractant. If you like the smell, it's an attractant to you. You dislike the smell, it's a repellent to you. Now it also happens that too much of the same deodorant or perfume I might not like, right? And we often say that my nose got sensitized. That's because too many odorant molecule came and my receptor is now clogged or receptor is oversensitized because of that. So oversensitivity can also be problem, right? So here also you have the numbers which are decoded, right? So what I want to tell you is that in a simple mechanism, simple in the sense we only say that we are smelling something, you have a very complex interaction going on. You have a lot of decoding, you have the integration and differentiation of these different kind of smell which come together, and then ultimately you come up to a decision. Okay, so you can have a garbage smell and a normal smell right next to each other, and you need to integrate and differentially understand. Basically your brain needs to understand these and then respond to accordingly. If you are closer to a garbage, you start responding in a different manner. If you cross some meters and you by the time this has been processed, you have reached to a better place, then you have to respond accordingly. So all of these integration differentiation takes place within a very simple term of smell, right? And I hope my questions give you more of these technical terms and you are able to understand them better, right? Okay, I hope I did a decent job to revise and help you recap the topics. And now I'll give you some questions, and hopefully we'll cover majority of the question. In case we don't do, the version will of course be available to you with the description or the notes having the description for the answers and the question. Okay, okay, so let's just begin so that I have spoken enough and I don't want to make you bore much. Okay, let's do more questions so that it becomes easy for you. The first question here is, the stretch reflex is an example of what type of reflex are? Polysynaptic, monosynaptic, autonomic, or inhibitory, right? Next one, what is the primary purpose of the stretch reflex, right? So why do you have this entire stretch reflex? Do you want to maintain muscle length and posture, generate voluntary movement, or the others? Okay. I still don't understand, how does perception change when there are same amount of binding? Okay, I will come to explanation. Just let people answer the question and then I'll maybe explain you the thing. Okay, because I don't want to disturb when someone is trying to answer, but yes, I will come back to you. Don't worry about that. Okay, I see one response. Others can also type in the chat box. Okay, 1B, 2B. Okay, just give me 1 minute. I need to sign. Yeah. Okay. Let me first give the answer and then I will reflect on the question in the chat box. So the first one is monosynaptic. We have discussed this already. And the second one is also muscle length posture, which is something that we have discussed so far. So I'm pretty sure that makes sense to you. Okay. Now coming to this question, which is, I think, very simple to understand. If you just remember the similar kind of question in the previous sessions. Okay. Right. And it just shows, I have been telling that this is a very smart crowd. So you guys get the answers very quickly. So you people are too good. Okay, now let me give you the question's answer. So how does perception change when there are same amount of binding? When you say same amount of binding, I am guessing that you are just saying that number of odorant molecules bind to the receptors. Now, we are only looking at the numbers, but along with numbers, there are several other things that come into play when there is a receptor ligand interaction. What are those? Think in terms of the binding affinity, think in terms of the binding speed, and think in terms of the competition between different receptors or different molecules, right? So what all these mean? What I simply meant was you have one receptor, it can bind to different odorant molecules. Why? Because let's say odorant molecule. So if you have, how do I say? If you have 1,000 receptors and you have 1,000 odorant molecules, it goes very good. But if you have 2,000 molecules, do you want 2,000 receptors? No, that's not how it functions, because it means every time a new molecule comes, you either go smellless or your system says, oh, I will first mutate and get a new receptor for it. That's not how it works. So what happens? Receptors are generally specific to one type of odorant class. So it can bind to different odorant molecule with different affinities. With one molecule, it will bind very strongly. With the other, it will bind with less affinity. What does less affinity mean? Just in case you are not aware, uh, an enzyme or a receptor and ligand binding can be reversible. Okay, it's reversible. So if it's weak binding or weak affinity, it will bind and loosen up quickly. If it is a strong affinity, it will bind very tightly. Okay. Now, why does weak affinity comes into play? Let's say there are three receptor molecules. Oh, sorry, there are three odorant molecules, and there is this one receptor, A, B, C. It has very strong affinity to A. Okay, now there is a competition between A, B, and C. All three will come and try to fit in this loop of in the groove of the receptor. But because A fits the best, it will bind quickly because it can overcompete the other. Because the other two will bind, there will some. Just imagine in vague terms, there will be some space available, and the other molecule can approach as well. But whichever fits the best drives the other two out of the competition, right? So you have different molecules. Now, why am I saying this? Because all of these types, I just told you, right? Just binding space. So the speed of the binding, the affinity of the binding differs. And because there is a difference in terms of other such patterns, you have different perception, right? Because if it was one-to-one interaction, which is just 1,000 molecules, 1,000 receptors, that's how you bind it. That is not the case here. The 1,000 molecules will bind in different, we call it dynamics or kinetics. So you have different dynamics of the binding, and that's what determines the perceptual changes. So it's not just the number of molecule which is binding. You can have how quickly they are getting activated, how fast they are binding to the molecule, how fast they are dissociating with the muscle molecule. So all these different activities together sum up and give the perception. So if they are loosely binding, obviously they will get less activated than the other which is strongly activated. So this is something which also was asked in previous session. All the mechanisms that we will read in future or in the past that we have read, remember, it's not a one directional or one dimensional phenomena. It's so many information together. If you remember, professor used words like differentiation and integrals, and it just tells you that there is such a complex amount of information coming for the brain to process, right? Okay, so yeah, let's move on. If you have more questions, please put in the chat box. I will be very happy to answer. But in the meantime, let's do this question. So you know the sensory afferent neuron, right? Afferent is something which comes, like which goes out. In this case, the muscle. So which one is that neuron? And the second one says which neurotransmitter do you is released by the alpha. These are very simple questions to begin with, so that's why it's in the beginning. You're welcome. Please do put in if you have any other doubts. So which is a sensory afferent neuron and which is a neurotransmitter which is involved there? Acetylcholine and third. Okay. Anyone else with any other answers? Okay. I'll just wait if someone else has to answer. Can go ahead. Okay. Okay. Just a second. Okay. Let me give you the answer. So this and this, right? C for both of them. Now, I guess the second one was very easy. The first one is also because we have already seen about 1 alpha, right? 1C, 2C. Yes. Okay. We did this already, 1 alpha and yeah. Okay, let's move ahead, which is again simple. So the primary receptor involved in the stretch reflex, which is the one that receives the information, and which of the following muscle act as effector? So which is the in effector in patellar tendon reflex, which is the other name for knee jerk reaction or knee jerk reflex. So in knee jerk reflex, which is the muscle that acts, and in general in stretch reflex, which is the muscle that receives the information. Easy question for everyone, I think. Any idea? Especially about the second one, knee jerk reflex. Which muscles are there in your knees? What are the options? Hamstrings, gastrocnemius, quadriceps, and biceps. If you have used these terms, you will if you're a gym guy, you will definitely know which name is this. Okay, I've got one option. Anyone else? Even if you are not a gym guy, these are terms that everyone uses everywhere to these days. The first question is what? The receptor. Which one receives information and which one gives out the movement? No? No worries. Let me give you the answer. So muscle spindle, always remember if we are talking about stretch reflexes or any reflexes, we are talking about muscles. And the next one for knee jerk reaction, the muscle in the knee is quadriceps. Biceps are here, hamstrings is in the thighs. And gastro, as the word suggests, you will know where they would be present. Yes, great. B and C. Next one. This is about extrafusal and intrafusal muscle fibers. So extrafusal muscle fibers are responsible for what? And intrafusal muscle fibers are located where? So different kind of question for similar topics so that you get division of both the subjects or both the topics. So where do you have extra muscle fibers, extrafusal muscle fibers, and what do they do? And where you have intrafusal muscle fibers, and what do they do? So one of each in each of these questions. Any idea? Okay. Anyone else? Okay, so I think we are in already 1 hour into the session. I will try to cover as much as question, but do remember, if I can't, a lot of questions are from the other two systems also. So please do remember to check it out when it's available on YouTube. Okay, both of them will say B, and as I said, this is a very smart crowd. So this is correct. Extrafusal muscle fibers for generating force and contraction. It is present outside the muscle spindle, or it is present outside the layer of muscle spindle, and intrafusal are present inside, and this sends the muscle stretch. Okay. Next one. These are on the two neurons that you have: gamma motor neuron and alpha motor neurons. So where are they present and what do they do? So the whole idea here of mine was to give you similar topics. Can you please elaborate once time again, intra and extra? I will come back. Just answer this question because they are related topics, and then I will tell you about intra and extra again. So yeah. So you have two, I told you, muscles and neurons are interconnected. So we have done about muscles. We are doing about neurons. 1C, 2B. Okay, C and B, right? So your alpha neurons are for the alpha motor neurons are in the extrafusal muscle fibers. Gamma motor neurons are to regulate sensitivity of the muscle spindle by contracting intrafusal fibers. Now, as someone asked, let me just quickly tell you to give you a mind map of what's happening. So you have extrafusal muscle fibers and intrafusal muscle fiber. For the timing, I will tell this as extra and intra. Okay, and you have three kind of neurons: sensory neurons, and then you have gamma motor neurons and alpha motor neurons. Now, what you need to remember is the whole idea here is to sense there is a distortion in your muscle length, sense it, take it to the spinal cord, process the information, and then come back to the same muscle to return or to come back to the original position. Okay, so who does this sensing of stretching? Intrafusal muscle fibers, which are basically present in your muscle spindles, right? So from intrafusal muscle fibers, along with this, the neurons which pick up this information are the sensory neurons. So your sensory neurons are in intra. Intra, you can remember it is inside. It is sensing. It is here basically. Just imagine it's here, right? It's the one who's sensing. So it senses a distortion, takes the information via your sensory pathway to the spinal cord. Spinal cord says, oh, there is a stretching. This needs to be contracted. Information goes to the motor neurons. Why motor? Because you have to know the movement now. Movement like movement of knees, right? So this is a movement. So the motor neurons. So the motor neurons, which are the alpha neurons, come back to the muscle. Now has to activate another muscle fiber so that it does the activity. This is the extrafusal muscle fiber. Extra and intra in terms of the opposite interaction it does. Intra because it is inside and it is sensing, and extrafusal is doing the opposite work. So opposite of intra here is extra, it is outside. So extra, it takes the information from the spinal cord. Okay, muscle needs to contract. So these muscles start contracting, and alpha motor neuron help him giving this information. The other motor neuron, which is present in the intrafusal muscle fiber, when this contraction is happening, it regulates the sensitivity of that intrafusal muscle fibers. So the whole idea is to sense an information, take it to spinal cord, get back information, and contract. I hope that was easy, or that was I could help you with the question. Okay, if not, you can use the chat box to ask questions. So we did alpha gamma. There's one more question on gamma just for your understanding. So we already did what it does. Just in other terms, what would you say? Why these different kind of questions? Like I always say, this is more of a course where you need to be careful about your technical terms. So that's why these different kind of question on the same topic. Okay. See, anyone else? Easy, right? These are easy questions. Pretty much. See, you are contracting the muscle fibers to maintain the spinal sensitivity. Okay, let's now start so moving away from this anatomical kind of questions. So read the question properly. We are talking about stretch reflex and we are talking about speed. Which one would be the correct answer here? A? Okay. Anyone else has any other option to give? Think. Okay. The cue for you to understand speed kind of question is what is the path, how long is the path the information has to travel? And path is determined by neurons. So ultimately, it boils down to in stretch reflexes, how many neurons are acting or how many synapses you have to cross. That is the key. That is the cue that I could give to you. Think, think in terms of monosynaptic and polysynaptic and the speed. Anyone wants to take a guess? You can just take a guess. I'm not going to argue on it. Okay, so I've got one response. Anyone else? It's okay. Wait, hold on. Yes, that's correct. Your answer is correct. But what is the answer to this question? If it is monosynaptic, that we know it is, what does monosynaptic do? How is the speed of monosynaptic? What do you think? Speed is fast or speed is slow? To everyone. So stretch reflex arcs are monosynaptic. So monosynaptic arcs are faster or closer, slower, faster. So which will be the correct answer here in this question? Okay. Okay. Let's see some. Right. You people have got to it from different routes, but that's you have got it correct, thinking in the correct direction. Okay. Just once quickly, I will tell you. One, yes. So your stretch reflex arc for people who joined late. Again, two type of reflection comes into your mind, right? Knee jerk reaction and you touching something hot and then moving your hand away. These are two different kind of reaction. The or reflex action. The first reflex action where your knee jerk reaction is considered, it is a monosynaptic reflex action, and where you're withdrawing your hand after touching a hot surface, that is your withdrawal type of reflex arc. This is a polysynaptic reflex arc. So the first difference is they are monosynaptic and the other one is polysynaptic. Here, how do you know what we are talking about? We are talking about stretch reflex. So stretch reflex are monosynaptic. Monosynaptic is basically one sensory neuron connected directly to the motor neuron. So you can see how this is a very small route to transverse in comparison to polysynaptic. What does it mean? That you have a sensory neuron, one synapse goes to the interneuron, second synapse goes to the motor neuron, just in case of one interneuron. So this has two synapses to cross, so it is a longer route, so it is slow, while the other one, which is your stretch reflex arc, has just a sensory neuron, synapse, motor neurons, muscles. So this is a smaller route to cover, so this is fast and autonomic. Okay, that's the logic behind this question. Okay, right. Now, in addition to the agonist muscle contracting, what happens to the antagonist muscle in the stretch reflex? Agonist is the one which is acting with in terms of your the reflex. So antagonist is something which is not directly part of it. So what do you think will happen to that? See? C, right? Wow, that's extremely smart crowd, right? Okay, so just remember, even if we say it's agonist and antagonist, so let me first explain these terms just in case you want to explain it in formal terms. So agonist muscle is the one that produces a specific movement, right? And is concerned with the entire arc. So in knee-jerk reaction, your agonist will be quadriceps, the knee muscles, right? Antagonist is a muscle that forms the exact opposite of the agonist. So in case of knee-jerk reaction, remember the antagonist is your thigh, your hamstring. Okay. Now, what does a body in general want to do? It wants to bring your body to normalcy. So what happened in your stretch reflex arc is there is a stretching of muscle and you want to bring it back to normal. And the antagonist wants the same because the antagonist believes like, okay, you want to do this for the betterment of the body, so I'll also contribute. Just think in these funny terms, it will make it make sense, right? So what does the antagonist do here? So agonist has to contract, right? Your quadriceps have to contract. So what the antagonist does is it makes it easier for quadriceps to contract, and that's why it undergoes relaxation. So it goes reciprocal inhibition. So if that is activated, it is undergoing inhibition. So that forward motion. Yes, sure, I'll do that. I'll give the next question and I'll type in that here. Right. So your muscle in the knee has to undergo contraction. So your hamstring, which is the antagonist muscle, will say, let me make it easy for you. Let me do the relaxation so that you can contract, right? Think of it in terms of pulling of rope, right? So just try to imagine I am the agonist. I need to pull the rope towards myself. Now, antagonist is, let's say, my friend on the other side of the rope. We both want the same thing. We both want the rope to come to the better this side so that things come back to normalcy. So I will pull. Now imagine if the antagonist, which is my friend, also pulling the rope towards the other way, it becomes more difficult and the distortion of the system is higher. So what the friend will do, it will let the rope loose. It will leave the rope. As soon as it leaves the rope comes towards me easily. So it makes the entire process easy for me to do. So that's exactly what antagonist is supposed to do. Antagonist is supposed to aid the agonist in the entire process. So it relaxes. It is a reciprocal inhibition. Right. Okay. Next question. When a muscle is stressed, the muscle spindle does what? Simple questions, repetitive questions, yes, but gives you, uh, like solid understanding of the topic. Okay, I have put my email ID in the chat box. You can avail it whenever you want. Also, this Teams link, You can use this link to join the meeting even if there is no class, and use the chat section to put on your doubts. This window is also available to you. You can put your chat boxes there, information there also. Like I will keep checking it in the week. So if you have any doubt, you can reach out to me via this also. Right. Yes, coming to this question. So if your muscle is stretched, which is basically the entire beginning of the reflex arc, what does it do? Just remember it like this. Anyone else? This is a damn smart crowd. Just a second, I'll just show the answer. You know this already. It sends afferent signals to the spinal cord. It begins the entire signals. Afferent is something that moves out. So it moves it out. Okay, now listen to this. At the neuromuscular junction, switching to the next topic, muscle is dash synaptic, neuron is dash synaptic. Synapse is basically the connection between neurons and muscles. So which one is what? Which one is post and which one is pre? Pre is in front, post is after. Think of the neuromuscular junction. Think of the neuromuscular junction, and then think of the muscle. Think of the neurons. Think of the synapse, and then think which is pre, which is post. Post is next, pre is before. That's how the normal terms are. Do you want to just visualize once again? Pre means post. I mean, pre means, or you can imagine in terms of pre means has happened. Post is something that will happen. Post is later, pre is before. Do you want to change your answer? Or can you, when I say change your answer, I know that you have already guessed that one of the answers is wrong because I have got C and D as an option. So without telling you which one is correct and which is wrong, do you want to reimagine your answer? Okay. I guess I got the wrong vibe there. The answer is post and pre. Okay, let me just show you why. I think this requires your visualization. Wait, hold on. Okay, this is your neuromuscular junction, right? For us neuromuscular of transmission, this thing is later in the cycle, right? It will interact with muscle later. First, it will interact with the neuron end. So the pre is neuron end and the post is your muscle. So I hope I didn't confuse you, but I wanted you to imagine better. Okay. It's okay if you, but your initial thought was absolutely correct. So yeah, I'm sorry. Where did it go? Here, yeah. So your muscle is postsynaptic and neuron is presynaptic. You were thinking in the correct direction. And so answer D, no? Muscle is post, neuron is pre. Yeah? Okay. What I just did was I think it was Pavitra. I'm sorry, Pavitra. I didn't mean to confuse you, but this is something that is very common, where you are telling an answer and you are made to think again. You were not the one I was trying to, like say you're wrong. You are correct. So don't get, like not get confused listening to words. Just when I when I made you imagine, also I was telling you the same thing. Pre is something that you will interact faster. Post is something you will interact later. So. Just be attentive to what you are listening, because you answered these. I'm imagining your thought process was absolutely fine. So just rely on it and be more concrete about it. Okay, we're doing great. So that's why I'll not consider the next option. Okay, so this is, I think, very easy. If we are talking about neuromuscular junction, which is a neurotransmitter we are talking about. This should be very easy to answer. People? Yes. Right. Okay. Acetylcholine. Yes. So now, if you are acetylcholine, you know, then what event triggers the release of acetylcholine? Basically, what triggers the release of the neurotransmitter? How does the neurotransmitter get released from the neuromuscular or neuron end? Read it properly, okay? We are talking about localization and the type of ions involved. Anyone else? Exactly. Oh, sorry. You said B. No. That's what I was trying to tell you. Yes. It's good you answered incorrect so that I can tell you again. There are three things happening in the neuromuscular junction. Remember, excitation and contraction coupling. When I am talking about acetylcholine, I am talking about synapse. So at synapse, how does your acetylcholine comes out? If you remember that, it was the fusion of the vesicle and the burst of the vesicle and the vesicle coming out with the neurotransmitter. And that was happening because of calcium ion in the neurons in the presynaptic terminal. This muscle is about muscle contraction. The calcium that you are confusing is with sliding filament theory, which is basically actin myosin movement. The question here was about neuromuscular junction acetylcholine receptor or rather acetylcholine release. So both of these are calcium. And you people wrote B, which is depolarization. Depolarization of muscular membrane, or basically muscle, led to the ultimate release of calcium for the actin myosin to interact. So the sliding filament theory takes place. That is for contraction. But when you are talking about your neurotransmitter release, it's the calcium vesicle, calcium influx sarcolemma, which is leading to the fusion of the vesicle. Okay, don't get overwhelmed by the topic. Just be calm, read. You should be able to do it. This was not difficult. I keep telling people on in my sessions, look at cues in the answer. What was the cue here? The cue here was acetylcholine. Where is acetylcholine? In the synapse. Now, when you know it's synapse, something which has to do in the neurons has to release, because something has to happen in the neurons so that acetylcholine is released. What is the only option about neurons here? This one. This is muscle, this is muscle, this is also muscle. So that can also be that is also a way for your checking the answers. Okay, so if you just, I'm sure you knew the answer. Just read the options very carefully, because these would be in your exams, and these are the, so most of these questions are from the assignments of previous years. So be mindful of what you are answering. Okay. Okay, now muscle contraction, we have moved to contraction. Okay, now when your muscle contraction, in case of your muscle contraction, calcium ion are there. Which one senses calcium ion? So basically, imagine what happens after calcium ion is there. What happens after calcium ion is released? The entire thing. I have one response. Anyone else wants to give it a try? See, okay. Okay. See, okay. So the answer will be B. And okay, should I show you the slide related to this? Wait, hold on. Is there a slide related to this? Not exactly. Okay, fine. No worries. So, okay, not calmodulin, but troponin C. And yeah, I'll come to it. Yeah, I will first explain why troponin C, and then I will explain what is calmodulin doing. So if you remember what I told you about, we have discussed contraction, we have discussed muscle contraction. Remember actin myosin interaction brings it closer to each other, so you have contraction of muscle. So your muscle contraction is basically actin myosin interaction. Now, if you remember how actin myosin interaction takes place, is because of the movement of tropomyosin, right? And why will tropomyosin move? If tropomyosin has to move, your troponin has to interact with someone else, because tropomyosin are sitting on the actin binding place. So your troponin interacts with calcium, and this calcium takes away the troponin C. Tropomyosin gets free, tropomyosin moves away, actin myosin starts interacting. That leads to your contraction. And the whole idea about your excitation and contraction coupling is that your actin myosin filaments will act together, right? Now, in its entire process, where does calmodulin come into? Calmodulin is activating your myosin light chain kinase, right? So that's way below. It works with the myosin. Calmodulin works with the myosin. But the question here was, let me just tell you again, it says sensor for calcium ion, which is basically any molecule or the protein that reacts to calcium, which basically means they interact with calcium. So ones that interact with the calcium, or the one that is responding the first to calcium here is troponin C. Okay, and troponin C, then tropomyosin, then actin myosin, and then when actin myosin are interacting, that's when your calmodulin comes into play. The question was sensor of calcium ions. Sensor means someone who detects the presence. Okay, all right. Next one. Huh. Easy. The role of sarcoplasmic reticulum in muscle contraction is to do what? It's basically the previous part of the last question. What happens before the calcium ion thing happens? B. Anyone else? B. Okay. Right. So let me give you the answer, which is, of course, stores and releases calcium ion, as we already know. So there were, remember, two receptors. The receptor interacted, your sarcoplasmic reticulum got activated, released calcium ions, and then the entire thing of troponin C and actin myosin takes place. Fantastic. Okay, then I spoke about the receptor, so let's talk about the receptor here. So the ryanodine receptor in the muscle fiber is located exactly where? Now, how do you remember this? There are two receptors, so you need to be very careful about which receptor I'm talking about. Is it the other receptor or this receptor? And when I say this and that, I will just cover the questions related to this, and then I will explain the entire thing just in case you are confused. But for now, this receptor, ryanodine, where is it present? Okay. So someone said A, and then which is basically T tubule membrane, and then someone says sarcoplasmic reticulum. Let me tell you, you both are thinking correct because you both are thinking about the location which is associated with receptor. But one of you is thinking for the other receptor. The answer for rhinodine is sarcoplasmic reticulum. The T-tubule membrane is for the other receptor, which is this one, DHP receptors, right? So if you're depolarized, so when your excitation contraction coupling takes place, when there is depolarization of T-tubule, what does it mean? What does it do? So out of these four, if your T-tubule is activated, or rather it is depolarized, then which interaction follows? Does it activate the interaction of the two receptor, or does it activate these voltage gated channels? Okay. Again, I've got two responses, and this time the correct answer is this one. Depolarization leads to the two receptor interaction. This ATPase, myosin ATPase, and acetylcholine esterase, this is nothing at all. This fourth option is just out of the bounds. The first question, this ATPase here, this ATPase is the reason for the depolarization. Depolarization does not activate this. Your sodium potassium pump is activated, or sodium potassium channel is opening up, first of all. That leads to the movement of sodium. That is why you have the depolarization. This is after this, right? ATPase is secondary. ATPase for your repolarization. This is later, if you remember that entire topic. But for now, what is important is your depolarization happens because sodium is moved in. Myosin is next, later, and that too is ATPase later. What is the immediate next thing after sodium has started moving in? You have your dihydropyridine receptor, which gets activated, which binds to the rhinodine interaction or rhinodine receptor, and then they together let your calcium release, right? Okay, so there are two more questions, and then I will explain this entire thing in case you are confused. So your DHPR receptor, what can you tell about it? In other terms, what do you tell this receptor as? Anyone? Fantastic. Yes, they are voltage sensors in T-tubules, right? What is the other thing that you can tell them? One was voltage sensors. What is the other sensor? Or what is the other term for these? Okay. Anyone else wants to answer? So are these receptor potassium channel? Are these receptor chlorine ions, or are these receptor calcium sensors? This one is simple, right? This is basically sensor. So let me tell you the entire process again before the next questions will be about your the other systems, right? Just for your revision, your muscles got activated because of acetylcholine. Okay, so let's start from acetylcholine. Acetylcholine in the synapse binds to this receptor on the membrane of the muscle. As soon as your acetylcholine binds, your sodium channels open up, sodium movement starts taking place. As soon as your sodium moves, there is change in the ion concentration of sodium in the muscle. This leads to depolarization. Now, when your muscle is depolarized, what is the first thing that happens? Your action potential will travel to sarcolemma. And in sarcolemma, you have T tubules, right? In the T tubules, you have DHPR receptor. This DHPR receptor will say, oh, there is an action wave. I am activated. I will now interact with my next partner, which is the next receptor. These two receptors interact. And then these two receptors further carry the action potential to sarcoplasmic reticulum, which leads to release of calcium. And from calcium release, we have read about action myosin potential attraction and sliding filament theory, basically muscle contraction. So that's how you connect these cycles. So my suggestion to you is read and try to visualize, and this will become very easy for you to understand. Okay. So we close the skeletal muscle system here, and because it will also be on the recorded thing. There might be, most probably there will be an additional class before the completion of all the 12 lectures that we have, which will be an extra class, which will not be on a Tuesday, which will be on some other day if it is there. What the idea of that session is to basically give you a recap of this entire course. Okay. So skeletal muscle, I will again discuss in that lecture, but very briefly. Okay. For the time for I'll come back. Yes, I'll tell you. But basically, it's in my Gmail. I mean, the email ID that I told you. It tells you where I am from. Yeah. So in the extra class, all these systems that we are discussing, I will have 2-hour session probably, where I will just give you more questions which are not repetitive of these questions. Okay. So you will have skeletal system once again, but for now, for the remaining four classes, we will not be discussing skeletal muscle anymore. Right. Okay. Now let's move to hearing system as much as we can cover. So the first question here is the auditory cortex in the brain does what? And which is the organ that converts the sound wave that we see into the nerve signals which goes to the cortex? One B, 2B. Do you also want to attempt? Any guesses? You can take a guess just in case. That will help you start thinking about the topics. Okay. Okay, let me give you the answer. So it's B for both. So the first one is that the auditory cortex, which is present in the temporal lobe in the brain, that is the ultimate interpretation center. So it gets all the information from the nerves, auditory nerves, and they process and interpret the sound signal. When I say interpret, I basically mean decoding the sound in terms of its pitch, in terms of its amplitude, in terms of its location. Okay, and then perception and giving the responses. And the next one was which organ is responsible for converting the sound wave into neural signal, which is basically your cochlea, which is present in the inner ear. So I told you, right, this is a fluid-filled structure. It has perilymph and lymph. So there you have your sound waves which come into the inner ear, and through the action of hair cells, which I told you, as your membrane moves inside because of the movement of bones in the middle ear, it creates a wave in the inner ear. In the inner ear, wave gives movement to the basilar or basilar membrane, and that membrane movement causes the hair cells to bend, and as the hair cells bend, they generate electrical impulse, which are carried by the nerves and carried to the brain. Okay, so that's the entire process of taking the information. Okay, so next one. The auditory nerves, they transmit signal from cochlea to where does it actually take? Which place do you tell final, like we have discussed about spinal cords, right? So in terms of auditory nerve, which one is the final location? And the second question is, I think, very easy. Any guesses? The second one is easy, I know that for sure. The first one, okay, received response from both of you. Okay, second one, I'm not going to discuss. We've already discussed it. The first one is basically, so your unlike your stretch reflex, which goes to spinal cord, which takes decision, the taste, the smell, the words, sounds, all goes to brain. Different parts of brain that we had done in one of the sessions previously. So these sound goes to temporal lobe or the auditory cortex, and the information processing of sound takes place in the brain. Okay, don't get confused, not in spinal cord. Now, remember I told you two terms, kynocelia and stereocelia. So the next two questions are for the differentiation of that. So if you have hair cells in ear which have one long hair, which type of hair cell is that? Stereo? Okay, well, that one is kyno. So the other one, which has multiple short hair, is stereo. Okay, so just remember, hair cells which have one long is kynocelia. Hair cells which have multiple short hair are stereo. You can remember in this in term of if you know about stereocelia or stereo cells, which are basically tandemly repeated, and so you can just remember it like this, that these are tandemly repeated and they are multiple hairs. Okay, now we have discussed about stereocelia and kynocelia about structure. Let's look at their like function. So a stereocelia, what does it do, and a kynocelia, what does it do? And this will close in on your topic of stereocelia and kynocelia. Two B. Okay. Any other guess? 1B, 2B. Okay. Let me show you. Let me give you the answer. B, B. Like I said, I don't have to even like wait from long. So like I said, stereocelia are the shorter here. So they detect the mechanical movement from sound wave, easier for them to do. And kynocelia are the long microtubule, which is their long hair basically. And they assist in hair cell signal transduction. Okay, so they also detect. So the difference between them is in their structure. Okay. Now, hair cells, they also generate action potential. They have to, because they have to pass on the information to nerves. So what do they do? When do they, so how does it actually get generated? B? Okay, anyone else? I mean, I do have any other option answer. You can try attempting this. Okay. All right, let me give you the answer. So like I said, so these hair cells that we see, right? Their main feature of them is to bend. Okay. Now, when these cells are bending onto the membrane, there is these sodium channels. Okay. As the cells bend and give pressure on them, they open up and the sodium ions move inside outside, leads to depolarization, action potential generation, nerves get activated. So all of, see, the, how do I say, the mechanism of generation of action potential is constant. You need sodium ions to move out and in. How does the sodium ion move is the thing that you need to remember in each system, because you need nerves to conduct. Wherever nerves has to conduct, it has to generate action potential. So there has to be movement of sodium. How does that take place in each of these systems is what you need to remember and pay attention to. Okay, so that's why the basic units, muscle, actin myosin filaments, nerve conduction, these are basic units. These, once you remember, you'll not have to think twice. The beginning of each of these units is what you need to remember, and just try to focus on them. And as much as you read and try to solve question, it becomes easier for you to do that. So again, mechanosensitive movement of your something on it. Sensodium channels are activated when, that's what I just told you. So in other question, other terms, the same question. Yes. Sorry. One, right? So your stereocilia will bend towards your kinocilium. Multiple short hair bending towards long one bending towards your sodium channel. Sodium just activated. Okay, very different kind of question. I just wanted to tell you how does sound change. So you can take a guess here. If there is a 1 decibel sound, so how much uh, does this represent an increase of sound energy? This is just for your, like just an information that I want to give you. So you can take a guess quickly. If you know the answer, well and good. If you don't, it's okay. I will explain to you. Just take a guess. See? Okay. Okay. So nice. So the answer is 1.26. Now let me explain you what happens here. So 1 decibel. So dB full form itself is decibel. Okay, just you need to remember first this. Now this decibel is actually a logarithmic unit. Okay, and logarithmic unit of expressing ratios of power. Okay, so let's say you have, so the basic idea here is, let me get a pen for you. So your dB, which is your decibel, is basically log of something, power 1, power 2, log of power 1 by power 2 into 10. 10 deci, because I'm talking about 10 decimal. Like I'm talking in terms of 10 times of the log 10 of these values. Okay. Sorry. So this is log 10. Okay. So when you have a 1 decibel increase, your power increases by 10 to the power 1 by 10. Which, so that is somewhere around 1.25 something, rounded off to 1.26. Okay. So what I wanted you to imagine is, even there is a 1 decibel increase, your energy of power increases by 1.26 times. Now, why I say this is important? What is the human hearing range? Any idea? Okay. If not, so your human ears, we can hear a range of frequency from 20 hertz to 20 kilohertz. And these terms, so by the way, dB is actually intensity of the sound. Okay. So if I say 0 dB, it means it's a very, very minimal sound that you are hearing. If I say 120 dB, it means it's a huge sound. Okay. Now let me just give you sound and try to imagine. So if I tell you 0 decibel, it means there is a not even a whisper. It's just sound that you can't hear. Okay. A whisper, which is basically when you want to talk in terms of code language, that is your 30 decibel. And 60 decibel is what I am talking in right now, normal conversation. Now, when you have 120 decibel, which is basically double of this decibel if I'm talking about, this is basically the rocket speed, or not rocket, but the jet speed that you had, the flight, the aeroplane when it moves ahead. So this is 60, that is 120. And just imagine the power of that sound. If 1 decibel is 1.26 times more power, that is 60 decibels higher. That sound of the aeroplane is so much more powerful. So I just wanted you to imagine the range of the power. How decibel log formula? Okay, maybe I haven't written it clearly. Hold on. Okay, give me a time. I have another slide. I'll put it up in that and then show, because it's difficult for me to write and show it to you here. I can use whiteboard maybe. I have never used it actually. Okay, wait, I don't want to complicate things here. Just give me 2 minutes while I'm giving you the next question. I'll show it up in the slide and tell you. Okay. Okay. Meanwhile, you try to answer this question. I will get that thing on the slide for you. So the process by which different frequency are encoded, which is basically decoding, along the cochlea. So what happens when different frequencies are decoded? So you try to answer that. I will show you this quickly. See? Okay. Anyone else? I can't recognize this question. Which question? This question? Basically option. Yes, pretty much. That's because I wanted you to have different ideas. So not everything I could cover up while I was revising the topic. So there are some, so you will often find questions in my session which cover up additional topics on these or subtopics of the main topic. That's why these questions. So you are free to take a guess, and then I will of course explain the answer to you. Right? So let me give you the answer here. Like someone said, it's place coding. So the question here was the process by which process helps you encode different frequency along the cochlea. So cochlea will have receive all these different vibrations, right? And then it is supposed to break down the frequencies that are coming, and that entire process is called place coding, right? Now, what does it do? What is place coding? So you define place coding as a process where different frequencies of sound are encoded by the location of the maximum vibration. So whichever, like you have sound waves coming, wherever the maximum vibration is there, you start coding accordingly. Okay, this one is giving the maximum vibration, this is giving the minimum vibration. So how does it maximum and minimum vibration happen? Remember frequency. If you have a high frequency sound, you will have more vibrations. Now in comparisons, if you have high frequency, they will give the maximum vibration, which will be near the base of cochlea. That's how the frequency of the, if you remember, professor had spoken about. If you have high frequency waves, so sound waves which have high frequency are repetitive in nature. So the high frequency waves are generally, their vibration remain at the base of the cochlea. The ones which are low frequency, they are at the apex. This depends on the waves also, amplitude also. All of these sound mechanics are in play here. But for the time being, just remember, if you have a high frequency wave, it is at the base of the cochlea. If you have a low frequency wave, it is the apex of the cochlea. And depending on where your vibration is in the cochlea will determine what is the type of sound that is coming. But what is it using to understand the type? The place of the vibration. So place coding tells you the type of the sound. That's what it means. Okay. So the spatial arrangement of these cochlear movements or vibration lets the brain interpret what kind of sound frequency is coming to the auditory nerve. Base means inside or outside. So if you see this thing, wait. Base is basically here, right? At the base, it's this thing, beginning. Okay. So yeah. Where did it go? My slides are getting mixed and messed up. Right. Okay, now let me just quickly show you this slide here so that I hope you understand. So dB, this is what I was trying to show. I don't know if it's clear to you or not. dB, decibel, this is the formula that is used for calculating your decibels. 10 into log 10 to the power P2 by P1. Now, if your sound is increasing by 1 decibel, so your equation becomes like this. 1 decibel increase is 10 into log 10 P2 by P1. So if you remove this logarithmic, this change in power is basically taking everything to the left side, which is basically 1 into 10 to the power log 1 by 10, because this becomes 1 by 10 this side. So log 10 to the power 1 by 10. So P2 by P1 is 1.26 times. So P2 is 1.26 times of P1. Does this make sense? Why is it arranged like, which one is arranged like, I'm sorry, I didn't understand. Which one is arranged like what? So you meant why are they the waves, right? You can imagine. So if you have high frequency waves, okay, let me ask you what kind of so what is the difference between the wave mechanics of high frequency and low frequency sounds? Do you know? Yes. Okay, I will. So, okay. So, unfortunately, this is 8 o'clock. So this question is where I will stop. There are 15 more questions which I couldn't cover today. So you will find it in the recorded lectures. We will end this question with the question that was asked right now. And on that discussion, we will end this class. Okay, so bear with me. What I will do is I will open the ears for you. I might have got confused or I might not have been able to explain to you. So let me just open ears first and then we'll discuss from there. Okay. Just a second. Yeah, we can open here. Okay. So wait a minute. Let me come back to this slide here. Am I back? Am I audible? Am I audible? Sorry. Yes. So, ma'am, if they are present far, they can still handle sound, but low frequency can be travel far. Okay. Yes, I will just explain it once again so that I think we are getting very confused here. Let me just explain once again to you. Okay. Let me first begin with the structure. So, in the cochlea, so inside the cochlea, you have the basilar membrane. And this basilar membrane is different in terms of its stiffness. Okay. So at the base, which is near the oval window, here, here, here it is narrow and stiff. The apex is here, like I told, that it moves inside. So this one, which the one that you encounter at the beginning is the base. The one that you encounter at the end, will you move inside? I don't know how to place it as outside and inside, but you can remember in terms of movement from outer ear to inner ear. Okay, so from outer ear, as you start moving, base will come first, and then you have the apex. Let's just put it like this so that it's one singular term for the structure. Okay, so you have base, which is near the oval window, and apex, which is near the center of the spiral. Now, why these two difference? So the one that is at base, if you see here in the structure, it is narrow, and as it goes into the apex or begin towards the up portion, it becomes wide and flexible. Okay. Now, when your sound enters cochlea through the oval window, so it is traveling as waves. Now, the high frequency waves have different peak amplitude. Remember, I told you what kind of waves will come will help in travel, right? Now, based on your amplitude, based on the type of wave, the high frequency stay at the base because it is smaller, so it can travel here. The low frequencies can go up to the apex. Why? Why again? Because that is what I was trying to ask you. Do you know the wavelength? So your high frequencies have shorter wavelength. Low frequencies have longer wavelength. So if you're shorter wavelength, they are near the base. The longer wavelength, they travel further because they are long, so they can travel long. Just imagine they're traveling long, so they can reach the apex faster. So your high frequency have shorter wavelength, stay at the base. Low frequencies have longer wavelength that reaches to your apex. And that's how you have these two positioning. Did that make sense? Yeah? Okay. Right. Unfortunately, I won't be able to cover the remaining question, but let me just walk you through what I couldn't do today and just show you what you will find in your recorded or lectures or uploaded this thing. So we discussed till here. We have more questions on ears, and then there is application of hearing system. Then you have smell. So I have a lot of questions on smells so that you understand each of the part of smell better. And then the application of smell. And then you have assertion reason, which if you read and try to answer, you will get the answers very well. Okay, so all of these are there in the video as well with the answers. You can pause and answer and look into the correct options. The explanation wherever required will be there in the presentation that will be uploaded. So both of the resources will be available to you for your perusal. And yeah, that's about it. So we have to close the session today here. If you have any question, any more, put it in the chat box. I am always available on my mail ID or this inbox here. If not, then we see each other next Tuesday with a very interesting topic to discuss. Okay, so that brings us to an end of the session. Thank you so much.